Okay, first video new. It's gonna be, I think, the only new video of the unit for Unit 3. Diving into cellular energetics. I'm not gonna talk too long because I assume I'm gonna do the same little dive-in thing for the next video. Sorry. Um, so, what we are gonna be focusing on in this video is enzymes, their structure, and their function, and things that can affect those two things. So, main thing, first thing you need to know, enzymes are proteins. So they're just a specific type of protein, and they act as catalysts inside cells and organisms. So they're essentially just biological catalysts, where catalysts are things that speed up a chemical reaction without being consumed, and they typically work by lowering the activation energy of the reaction, which you can see here. Over the course of the reaction without the enzyme, it requires more energy to get started than it does when you have the enzyme there. But the overall change in energy is not affected. So you don't get any more or less energy out of the whole reaction that you're doing. It's just that it takes a little bit less to get it going. It's kind of like, it's not exactly like lighter fluid, because lighter fluid does get consumed. But if you're trying to start a fire, sometimes you have a little catalyst to get it going. There are two main parts, or two main things we're going to be talking about, two vocab terms. The thing that binds to an enzyme is called the substrate, and the place where the substrate binds to is called the active site. I'm going to be using those words a lot in the next little bit. Sometimes you'll hear this as the lock and key model or induced fit, where the lock is the enzyme and the key is the substrate. It has to have the correct shape in order to fit into the active site and for the enzyme to actually do its job. So, generally, what happens is your substrates will enter. It can be multiple different substrates, it can be one single substrate, but they will be attached to the active site, and then they will cause what's called the induced fit, where the enzyme actually changes shape a little bit to kind of lock in the substrates into position while the chemical reaction is going on. Then the chemical reaction happens. After the chemical reaction happens, the shape has changed once again so that the new products are actually kicked out of the enzyme and released. And then the shape of the enzyme goes back to its first step, ready to act on new substrates. So the new substrates come in, they bind, cause a shape change, the reaction happens, we get another shape change, the products leave, the shape goes back to normal, to the start, and then it's ready for more substrates. This can be done over and over and over again. Theoretically, it's infinite. In practice, our body's proteins do not last forever. They'll get broken down or degrade or denature or something else. But our enzymes are extremely specific. So they have a specific shape. The active site has a specific shape with specific chemistry that's only going to bind to specific substrates or things that are very similar to those specific substrates. This binding is typically through some weak interaction, so it's not a full covalent bond. It's ionic bonds. Remember, ionic bonds are weaker than covalent bonds. It could be hydrogen bonding, it could be polar, nonpolar interactions. Some form of weak interaction holds it in place. And we have an example of this enzyme specificity where if we have the two different types of glucose polymer, starch can be broken down by our amylase, whereas cellulose can't because of just the shape and location of these O ester bonds. A little bit on naming. Enzymes typically will end in ACE or ASE, and that is usually referring to the substrate they bind or the reaction they catalyze. So big examples: lactase catalyzes the breakdown of lactose. ATP synthase is the enzyme catalyst for the reaction that synthesizes ATP. So they try to help you out with the naming a bit. There are a few things that can affect how well an enzyme works. The first thing you can do if you want enzymes to work faster is just increase your substrate concentration. Add more reactant. At a low substrate concentration, you can see all the enzymes are not actively working. At a middle ground, it's a little better. At a very high substrate concentration, all of the enzymes are fully engaged and working all the time. And so you'll see that you approach this saturation point, this asymptote, where the enzymes are all saturated, they're all taken up, they're all actively catalyzing a reaction. And so the only way you could actually speed this reaction up at that point would be to add more enzyme. On the other end, we can also reduce our enzyme activity or make it worse. This process is generally just called denaturation, which is when you just change the shape of a protein, causing it to lose its function. It can be due to temperature, pH, or some other environmental factor, but it's just a shape change that means the enzyme no longer works. Talking about temperature and pH, these ones are the ones that come up the most often, and our body uses the most often. So temperature, usually we can assume that increasing temperature increases the rate of a reaction. And this is true to an extent. Like you can see that the bell curve does continue to go up for a little bit. But you hit this certain point where the increased temperature actually changes the protein's 3D structure, and at that point you get a rapid drop-off in function. So our typical human enzymes, they work best at 37 degrees Celsius, which is our body temperature, but they can work at lower temperatures basically just fine. Even at 20 degrees Celsius, which is room temperature, they still function, they're just slower. Whereas if I went 17 degrees above human body temperature, I am dead. So you can generally be a little bit lower in temperature. You can't go higher too much, otherwise you just die. But there are organisms that do this. Thermophilic bacteria near volcanoes and underwater sea vents will have higher temperature enzymes because they are adapted to the environment that the bacteria live in. pH is similar. Most enzymes in general will work between a pH of 6 and 8. That's just the most common solution for things to be in. Water, remember, being 7. But we do have some exceptions in our digestive system. So we have pepsin and trypsin, which are stomach and intestinal enzymes. And our digestive system does have a separation of pH. So our stomach enzymes work best around a pH of 2. That is the pH that our stomach is at. And our intestinal enzymes work best at a pH of 8, which is, again, what our intestines are at. And this makes sense in a digestive sense. If we, for some reason, get stomach enzymes into our intestines, we don't want those stomach enzymes working and acting on things there. Same thing with the intestinal enzymes. If they find their way into the stomach, we don't want them to be active there. So because these two enzymes work in odd pHs outside of the norm, it lets us regulate when those enzymes are active and make sure that those enzymes are only active in places that we want them to be active, because otherwise they would digest our bodies. And we're going to do a lab around this idea. It's a fun one. Other things that can affect it are cofactors and coenzymes. There will be some that we talk about, especially in cell respiration. This is just some other molecule that has to attach to the enzyme in order for the enzyme to function. So it's attaching somewhere else that's not the active site. It can be near the active site. But these are also not consumed by the reaction. They're just there. Again, it's another regulatory step. It's another opportunity for us to regulate the activity of an enzyme. So inorganic ones can be things like metal ions, zinc, iron, copper, hemoglobin. The iron in your hemoglobin would be one. Coenzymes are often things like vitamins. I'm not sure if vitamin D is one of them or not, but coenzymes are just organic cofactors. So very, very slight difference. But the main idea is that they will kind of activate the protein. The protein is only active if it has its appropriate cofactors and coenzymes there. It can be one, it can be multiple. And again, the purpose is for additional regulation of the enzyme. I can turn it off and on much more easily if it requires a cofactor or a coenzyme. Then we have inhibitors. Inhibitors are just any chemical that will attach to an enzyme selectively. So it's not just like acid that reacts with everything, but it is a selective attachment that inhibits the enzyme from binding to its substrate. So it can do this in two ways. First being competitive inhibition, where it just physically attaches to the active site and blocks the substrate from getting in. This is how caffeine works. It binds to the adenosine receptors in our brains. That is usually what can make us start to feel sleepy caffeine binding them prevents them from feeling sleepy because it is out competing the sleepiness molecule. Then you have non-competitive inhibitors. These just bind the enzyme away from the active site, but cause a shape change at the active site that prevents the substrate from coming and binding to it. So the main difference is just where they bind, if it's at the active site or not. And these can also be reversible or not. Sometimes your inhibitor binds and it causes a permanent shape change. More often, that shape change is reversible from the inhibitor. And that is it for this lecture. Soon we're going to get into metabolism, then respiration and photosynthesis. It's a fun time. It's super exciting. But that's all, folks. I wanted to say yabba dabba doo, but that's Fred Flintstone. Bye.